My name is Chuner. I'm a PGY3 resident in general surgery, and I'm going to talk about gene regulatory mechanism of interleukin-based immunotherapy in pancreatic cancer. So my journey to this project is kind of twofold. I've been interested in fibroblasts. I did my PhD dissertation in reprogramming fibroblasts to other differentiated cell types. And I've always noticed that fibroblasts are not all alike. They're different, and they vary depending on their tissue origin, their tissue context, and the signals that they receive. And the second part leading me to this project was my experience as a resident. As I spent more time on surgical oncology, I got to experience firsthand how pancreatic cancer is still a very, very challenging cancer to manage. On the picture on the left here is a specimen of pancreatic cancer, and you can see that there's this remarkably dense fibrotic material, stroma, that is thought to be resistant to chemotherapy penetrance. And yes, and these patients, even with surgical resection and our best chemotherapy, the survival rates are low. And microscopically, when you look at this cancer, you see the cancer-associated fibroblasts are a key component of this. They have diverse molecular functions, and they have crosstalk with other components within the tumor microenvironment. So what makes a fibroblast a CAF, or a cancer-associated fibroblast? They respond to a various number of signals, and they become activated in this cancer context. And some of these signals could include inflammatory signals such as IL-1. Historically, CAFs are thought to be mostly pro-tumorigenic, but this was proven wrong. In the study published in 2014, this group looked at ablating fibroblasts and cancer-associated fibroblasts that express alpha SMA. And their hypothesis is that if the CAFs produces dense desmoplasia that's resistant to chemotherapy, then killing them would help. But that wasn't what they observed. They saw that the tumor were more aggressive and the mice died faster. So this proved that some CAFs have tumor restraining properties, and they're not all alike. So CAFs are clearly heterogeneous, and they have an impact on tumor biology. And it begs the question to figure out what they are. And indeed, a lot of recent studies from here at Stanford and elsewhere have identified multiple different subpopulations of CAFs, including myofibroblasts, inflammatory, antigen-presenting steady-state CAFs, etc., based on their function. So then I want to ask which CAFs are pro-tumorigenic, and what are the signals that drive these pro-tumorigenic states as an important avenue for potential therapy. I want to focus on one aspect of it, which is the crosstalk with the immune system. Specifically, IL-1 is a well-known cytokine that is best known for its role in the innate immune system, but it also is shown to drive pathogenic inflammation in pancreatic cancer. Interestingly, it activates this inflammatory phenotype in CAFs, which is mediated by JAK-STAT signaling. And our work here at Stanford had also confirmed this finding. We have immunohistochemistry showing the co-expression of CAF marker collagen as well as IL-1 receptor in human PDAC specimen. And on the right, also, we have a mouse experiment where mouse bearing pancreatic tumors were treated with IL-1 receptor antagonists, and which has an effect in reducing tumor weight and improving survival. However, the results here are mixed and not always statistically significant, which is in line with other studies, previous and ongoing clinical trials looking at JAK inhibitor and IL-1 blockade in treating pancreatic cancer. So there's still a need to pinpoint the genetic regulatory mechanism that's specific to this IL-1 driven inflammation within CAFs. So to answer that question, we sought to look at the multi-omic landscape of PDAC in both mouse and human. So we have mouse tumors that were controlled and also ones treated with IL-1 receptor antagonists, as well as IL-12, which is a interleukin with a strong anti-tumoral effect. We have several human specimens as well from patients with pancreatic cancer who underwent resection here. You've heard about ATAC-seq from Jen Parker earlier. This technology here is a slightly different version where we do multi-omic profiling, meaning we're looking at the ATAC-seq component, looking at the gene regulatory portion of the transcript of the genome, as well as gene expression simultaneously in the same cells. So this gives us a single cell resolution landscape of the tumor microenvironment in both human and mouse PDAC, and will help us answer looking for the gene regulatory signals driving these pro-tumorigenic states. So our mouse study shows some really interesting insights from transcription factor enrichment analysis. Just to walk you through how we do this briefly, we have RNA and ATAC-seq information from the same cells at the same time, which we use to perform dimension reduction and cell type annotation. Focusing on the fibroblast clusters, we can then ask what transcription factors motifs are enriched in the accessible chromatin. And strikingly, there are these NFAT transcription factors that are enriched in fibroblasts compared to the other cell types. These are nuclear factor of activated T cell family transcription factors, and they're known to have a role in T cell development as well as a number of other disease processes, including cancer progression. When we look at the NFAT motif activity in calves specifically, we see that it's decreased when the tumor was treated with IL-1 receptor antagonists. This suggests that NFAT might mediate this IL-1 driven inflammation in calves. Then we want to ask, is this something that we see in human as well? Well, obviously we can't do the same kind of experiments in human, but we do have the human specimens. And looking at this multi-omic analysis of human tumor samples of both RNA and ATAC-seq data, we can do the cell type annotation in a similar fashion, find the fibroblasts, and ask what transcription factors are enriched in the fibroblast cluster. So looking at transcription factors by both gene expression and their motif accessibility, we see NFAT again as the top transcription factor regulator for these calves. And these are enriched on both the gene expression and motif accessibility level. So this is a conserved finding across human and mouse. Focusing on the fibroblast cluster specifically, we annotated the subpopulations based on previously published calf markers and found that NFAT actually co-localized with a subpopulation identified as inflammatory calves marked by CCL2 expression. And we validated this result to make sure it's not a purely bioinformatics artificial finding. You can see here that NFAT C2 expression also co-localizes with IL-1 receptor. And this is confirmed by immunohistochemistry in human specimen on a protein level as well. And from here, we think that NFAT drives the pro-tumorigenic inflammation by calves. So in summary, we sought out to ask the questions of what are the possible regulators of this pro-tumorigenic inflammation mediated by calves. We did this by looking at the multi-omic assay of both gene expression and accessible chromatin in both mouse and human, and identify NFAT as a possible calf-specific regulator with the proposed mechanism in response to IL-1 signaling and driving ICAF identity. This is an ongoing project. We're still doing experiments to validate these findings. I'm excited to see what's to come, but we are really curious to see if NFAT's role is specific to calves. So we have some in vitro model to inhibit NFAT and assess whether or not its effect is specific in calves alone. We also have an in vivo model by intratumoral injection of an NFAT inhibitor in the mouse model of PDAC tumor to see if there's any effect in tumor weight and survival. The overall big picture here is that if NFAT drives this pro-tumorigenic inflammation, can we use it as a target for therapy? And the thought is, if we have these calves are in the pro-inflammatory state and we have a regulator identified, we could potentially inhibit them to reprogram them and reduce tumor progression. And this is in contrast to traditional approach of using interleukin or other immunotherapy could be a stronger and more specific approach, given that transcription factors are the ones that drive cell identity and orchestrate that. So I'm excited to see what is to come. As you know, as a clinical resident, I'm really busy, so I can't do this work by myself. I like to acknowledge others who have helped me. I'd like to thank the program and the department leadership for funding my research and allowing me to do it. And I'd like to thank Dr. Norton as well, who is my mentor for this project, who's been an incredible role model, as well as my other mentors, Dr. Longacre and Dr. DeLito. Michelle and Anna have been working on this project for months and months prior to my analysis, so I want to acknowledge their contribution as well. And Masa, Jen Parker, Deshka, Michael have all contributed to this. So thank you very much, and I'm happy to take questions.